All right, well, let's see. Here we go. I'd like to welcome everyone to, this is our first Polyploid webinar of the 2022-2023 season. We've got a full calendar all the way up until May. One of these, the aim is roughly to do one of these sets of talks on the first Wednesday of each month between now and then. And then in May 23, we have another Polyploid meeting that'll be taking place in Gainesville, Florida. Details are to be determined and shared still, but I was told to mention it, and that it's on the horizon, so to put that on people's radar. But today we have two really great talks from Corrinne Grover and Evan Forsythe that are paired up and are telling different parts of the same story. And up first is Corrinne. Corrinne Grover she's a research scientist at Iowa State University, working alongside Jonathan Wendell at the intersection between polyploidy and other phenomena such as expression, evolution, and domestication. Her research addresses questions in polyploidy using the model-ish genus Gossypium. I think we can call it Cotton a model for polyploidy. And her work could be classified as polyploidomics if we're still appending that suffix on everything. Her work is generally focuses on the genomic and transcriptomic outcomes associated with polyploidy and the evolutionary paths afforded by this phenomenon. And today she's going to tell us about some really cool work that she's been doing in collaboration with Evan Forsythe and other folks on cytonuclear expression accommodation in polyploid lineages. And with that, I'll let Corrinne take it away.
Okay, great. Thanks, Mike, and thanks all of you for coming. As you mentioned, my talk today is about cytonuclear expression accommodation and polyploids. Is that even a thing? But before we get into that, I just wanted to tell you guys about bonus content for this talk today. We all have been hearing more and more about AI-generated art, so I thought, hey, could that be used to facilitate scientific communication? And the answer is, For a novice user like myself, you can generate really cool images like the two you see and a couple you'll see in the introduction. But in general, for us novice users, it might take a little bit of maturity before we can really use it. So without further ado, let's go ahead and get into the meat of the matter. And we're going to start with a spoiler alert. So if you are looking for smoking gun TB1s or the answers to life, universe, and everything, this is not the talk for you. But if you're interested more in subtlety and complexity, and sometimes in somewhat, then this is the story for you. So let's talk about cytonuclear accommodation. And realistically speaking, it's not something that's just unique to polyploids. People have been discussing cytonuclear accommodation in the context of homoploid hybrids for a while, because the idea is that you have two different species that have been walking two different evolutionary paths, and they come together in this common nucleus, right? But only one of them gets to contribute the cytoplasm. And while they're walking those different paths, there is a certain probability that they will have acquired mutations in genes that interact between the nucleus and the cytoplasm. And there's a certain probability that some of those will become maladaptive when these two merge again. And so there may be preferences biased toward the parent that in the nucleus, toward the parent that has contributed to cytoplasm as well. So if we think about something like rubisco, where part of rubisco is encoded in the nucleus and part of it is encoded in the chloroplast. In the context of a homoploid hybrid or a polyploid, when those two come together, the paternal parent, typically, may have acquired mutations that makes this joining of these two subunits not quite optimal. And those are the things that we think about when we think about cytonuclear conflict. Now, that's common to allopolyploids and to homoploid hybrids, but of course, we all know that polyploidy adds an additional dimension for everything. It adds more problems, but it also gives us more potential solutions. So, of course, polyploidy doubles the entirety of the nucleus. But what happens to the organelle? Is it a 2 versus 1 problem? And the answer to that is no, that actually, this is work by Matt Jorfi et al. From last year, which shows that the number of organellar genomes actually does scale with ploidy. So maybe we don't have to worry so much about the fact that we're doubling the nucleus while only inheriting the cytoplasm from one. But the other thing we know about polyploids is that there's also other things going on. There's gene loss, and sometimes it's random, sometimes it's non-random. And those are the sorts of things that can lead to stoichiometry and dosage problems when certain genes are lost, but maybe not others, and you're still trying to function with this uniparentally inherited organelle. So a lot of this, so we really started thinking about this in the context of Rubisco, again, because it is one that is encoded partially in the nucleus and partially in the chloroplast. And these two subunits have to come together and work together. And about 10 years ago, Lei Gong and co-authors looked at this in cotton and then later in 4 other genera. And they found that, yes, in the context of Rubisco, there is evidence of cytonuclear conflict. And this is resolved potentially through biased homeolog retention. That is, the maternal homeolog is retained preferentially. It's preferentially expressed. And gene conversion also favors the maternal homeolog. So, you know, it looks like, okay, cytonuclear conflict, it's a thing. And, you know, we're going to start seeing this pattern all over the place, right? Well, we're in science, so not right. And follow-up research in Tragopogon, in synthetic rice and cucumber, and in the broader Brassica complex showed more conflicting patterns and no clear signal of cytonuclear conflict and or mechanisms to resolve cytonuclear conflict. And then, of course, we have wheat, which didn't use Rubisco. Most of the rest of these used Rubisco, except for the Shan 2020, which is a broad survey, and the Brassica complex, which is also a broader survey. We used MS1, which is another cytonuclear gene that confers fertility, and that showed the same patterns as Rubisco. So it's not just limited to Rubisco, but it doesn't always happen in Rubisco. And what the heck's going on? So what do we know? We know that cytonuclear conflict is a potential outcome of polyploidy, and it may correlate to how far diverged the diploids are. You can imagine that yesterday's diploids would not have had time to acquire conflict, but that ones that diverged millions and millions of years ago may have had more time, more opportunity to acquire conflicting mutations. And we know that expression biases are one avenue to resolve this conflict, although we see inconsistency in the evidence so far. So in our research, we wanted to address this from the perspective of expression evolution. So we asked 3 broad questions. One, is cytonuclear expression accommodation common both among genes, so Rubisco and all the other genes that are interacting or that are targeted from the nucleus into the organelles, and also among polyploid lineages? And then if we do see evidence of cytonuclear expression accommodation, does it take the form of maternal homeolog expression biases, whereby the maternal homeolog is preferentially used? Or is it the total expression level that's important? And should that be more similar to the maternal parent than the paternal parent, this being maternal expression level dominance? So our setup. Because we have seen conflicting evidence in different systems, we decided to use a setup that includes 6 polyploid species, which are listed over here on the right-hand side, that stem from 5 individual polyploidization events. The Gissippian ones here, cotton, are shared and represent 4 different genera. The polyploids themselves range in age from this synthetic peanut right here to the quinoa polyploid, which is about 3 million years old. So if expression cytonuclear expression accommodation is something that is temporally based, maybe we can see that by just dissecting these out by polyploid age. Also, as we just mentioned, that it may be a function of diploid divergence. So we also incorporated, to the extent possible, a small range in diploid divergences here. And we used ribo-depleted RNA-seq in order to evaluate expression writ large for the nucleus and actually for the organelles. And I'm not going to tell you the organellar expression results because that's for Evan. But what I'm going to tell you is what the nucleus is doing with respect to expression, evolution, and accommodation for those genes that interact with the cytoplasm. So our reference, I'm going to try and go through this quickly because I have a lot to go through. But I'll note that we do have to, our reference included both mitochondrial and chloroplast transcripts because, as I said, we're dovetailing this with an evaluation of organellar expression. And then we included nuclear transcripts, removing those nuclear transcripts that may have, that are too similar to mitochondrial and chloroplast transcripts. They may be things that just got misannotated as genes within the genome. And we also did not want them sucking down all the reads from the organelles themselves. And so you can see we have a range in genes in the genome from about 44,000 to 74,000. But because for the purposes of this experiment, we are looking at how the homologs are acting differently from one another, we restricted our analysis after mapping to only those genes that we could reliably pin down as homologous pairs, which also had a single copy in the diploid. And that cut us down by quite a bit. And more so for peanut and quinoa because the resources are not quite as well developed. But we did drop down in terms of the number of genes, and that's natural. Some of those could be for biological reasons like gene loss, and some of those could be technical reasons such as multi-gene families, where we couldn't faithfully disentangle paralogs from homologs. So with these homologous pairs, we partition those into categories. That is, those that do not interact with the organelles at all. And those form our background, right? So any homolog expression bias that is genome-wide, it's going to be incorporated in this background, same for expression level dominance. And with the remaining cytonuclear genes, we partition these into 6 categories. Those that are targeted to both the mitochondria and the chloroplast, those that are targeted only to the mitochondria, and those that are only targeted to the chloroplast. And then within those, we further dissected those into those that actually interact with products from the organelle and those that do not, because you might imagine that there might be greater pressure for those products that actually interact with organellar products than those that are used in the organelle, but are maybe not interacting. So now you're listening to all this and you're saying, okay, Corrinne, you just told me that you lost a lot of gene pairs, okay? So maybe what you're seeing is that you're having fewer homologous gene pairs, which is due to loss, which is due to accommodation through loss, case closed. And I had the same thought. So we actually evaluated this for the 2 genomes where it was possible. Again, the resources in Chenopodium and Peanut were not mature enough for us to do this. But if you look at biased homolog lost in the context of this background, the not organelle targeted, you will only find one category where there is an excess of parental loss and it's biased towards the maternal. And for these, for this and pretty much the rest of the slides that I'm showing you, we're conducting Fisher exact tests where we're looking at the distribution between these, how things are partitioned in the individual categories versus how they're partitioned in the background. And what we're generally seeing here is no, it's not homolog loss that is the reason for our reduction, as far as we can tell. And this actually corresponds to something that Joel Charborough and co-authors published last year, or actually, sorry, earlier this year, which is using 6 allotetraploids. There was mixed evidence for patterns of cytonuclear gene loss and evolution. So again, we're seeing kind of a mixed bag when it comes to cytonuclear accommodation. Well, what do we see in our expression data? So the first thing we're going to look at is expression level dominance. And the idea here is that for most homologs, mom and dad may not have had enough time to acquire maladaptive mutations, but maybe expression level differences. There are expression level differences between mom and dad that actually make a difference when you merge them two into the polyploid. And now you have to coordinate transcription levels between the nucleus and the chloroplast or the mitochondria. So if this is true, if selection is acting on the total expression amount, we should see more maternal expression level dominance. And I know that expression dominance in polyploidy gets confounded a little bit. So when I speak about expression level dominance here, what I'm talking about is where the diploids have 2 statistically different expression levels, and the polyploid is statistically similar to one parent, but not the other. And in that case, the parent that is statistically similar to is the one where it's considered dominant. And so we can compare cytonuclear expression level dominance to background, those in the not targeted category. And what we generally see is that in general, there's more maternal expression level dominance in the background categories. That's these global checkboxes here. And you can see global maternal expression level dominance in 3 species, and paternal expression level dominance globally only in 1 species. So it seems like maternal expression level dominance is just more common. But when we restrict our attention to those genes in cytonuclear categories, natural peanut and cotton Gossypium barbadense both exceed the expression level dominance that is globally displayed by those species, such that it's statistically higher or greater than that. And quinoa, which has had no global evidence of maternal expression level dominance, does exhibit some for the cytonuclear genes. But there's not too much going on in Arabidopsis or in the synthetic peanut. And also, interestingly enough, in the other cotton species, Gossypium hirsutum, that stems from the same allopolyploidization event, we don't see evidence of cytonuclear expression level dominance that exceeds what is present in the background. And we went in the paper, if you want to look, you can look at the categories because we partitioned this out by, whether or not it's going to the chloroplast or the mitochondria. But the summary is that there is excess expression level dominance in 3 species. It's in limited categories, and there's no real pattern to it. So here we're starting to enter the somewhat and sometimes. So let's look at homolog expression bias, because, okay, maybe we can start seeing some real patterns here. So if we think about homolog expression bias, when there's conflict, maternal homolog should be preferred. And this could present itself as either maternal upregulation and paternal downregulation relative to that seen globally in the background. And be prepared for the world's most complicated figure. This is one directly from the paper, and I don't expect you to digest the whole thing because it's a lot. But a few things that you should note is that the global bias in homolog expression is up here in the subscripts. So here are the species: Arabidopsis, the 2 peanuts, quinoa, and cotton. Only the synthetic peanut has maternal expression, homolog expression bias. The rest of them generally have paternal homolog expression bias. Then if you look through the categories, which are here in these rows, we can see in the individual cells, there are quartets here. And these quartets describe whether or not we see an excess or fewer genes than expected by global patterns that are upregulated relative to their parent, and upregulated are the top two cells, downregulated are the bottom, mom is on the left, dad's on the right. And like I said, totally complicated, but what you want to note are the patterns down here, where this is the example of what's consistent with cytonuclear expectations, and this is the pattern that is contrary. So in the pattern consistent, more genes than usual than in background for mom are upregulated and fewer are downregulated, and more paternal genes are paternal genes are upregulated than background, and more are downregulated. And still, that's a lot. If you want to read the paper, you can spend tons of time digesting this, but the summary is in the highlighting. So if you look at the yellow cells, those are the ones where the pattern is consistent with cytonuclear expectations. And the grayed out ones are where the pattern is contrary to cytonuclear expectations. And the white ones are just mixed bags. And so the takeaway from this is that some categories are consistent with cytonuclear expectations in some species, but we don't have any clear patterns regarding species or category. So again, we have a sometimes and a somewhat. So then we wanted to directly compare expression between maternal and paternal homologs. So the previous slide I showed you was upregulation and downregulation relative to the diploid parents. But now we can ask the question, is the difference in expression between homologs due to targeting? But we also have to include a term for the diploids themselves, right? Because we know that we generally see that expression patterns are inherited, at least to a certain degree, from the diploids themselves. And so when you create a linear model that looks at the difference in expression as a function of targeting and with respect to the difference in diploid parents, again, very complex figure that I'm not going to go through entirely, but I'll just kind of digest for you. What we see is that for each of the species surveyed, the targeting term, the difference in the diploid parental expression, and the interaction are all significant predictors of expression in general. And then if you look at the fixed effects for each of the species, you can see that all the categories have significant fixed effects in at least a few species. Sometimes the fixed effects are positive, red, sometimes they're negative, blue. But what you can see here again is that we're not seeing any consistent patterns with respect to the fixed effects in the linear model. And then if we look explicitly at the contrast between the cytonuclear category and the genes that are comprised of background, the not organelle targeted, again, sometimes we see significant contrast, sometimes we don't. Sometimes they suggest an excess and difference in the organellar genes. Sometimes they suggest an excess and difference in the not organellar targeted genes, or rather that these are closer in expression level than maybe one would expect. So again, we're seeing somewhat and sometimes here. So, oh, there we go. I forgot I had that. So targeting matters, but the patterns are generally inconsistent with respect to the categories and how they predict homolog expression. So given these conclusions so far, that expression level dominance, that homolog expression bias, that just the expression between the homologs themselves is not really showing us consistent patterns of cytonuclear accommodation, you might be thinking back to Rubisco, right? Because what the heck is going on? in Rubisco, at least for some things, we saw clear evidence. Well, did you see that here in your data? Well, we were not able to fully evaluate Rubisco in our data. In Peanut, we were able to pull out 6 copies of Rubisco, but due to the resources available, I can't tell if they're maternal or paternal. In Chenopodium, a similar problem. I was able to pull out 1 homologous pair, and that's it. For Arabidopsis and Gossypium, we had similar issues in terms of I was able to determine maternal versus paternal copies because they were assembled into maternal and paternal scaffolds, but we were only able to determine 1 homologous pair for each of these ones as well. So in Arabidopsis, we did not see significant differences between maternal and paternal homologous copies, but that doesn't mean if we dove back down into the weeds like Gong et al., that we wouldn't see the same patterns that Lay and others saw when they evaluated Arabidopsis. If we drop down to Gossypium, because that's another one that Lay looked at, Hirsutum, at least for the gene pair or the homologous pair that we could determine, it fits the pattern. So maternal is more highly expressed than paternal. And I can tell that just by the sign. This is a fold change, and the sign tells me that maternal is more highly expressed. But for Barbadense, it wasn't significant. Now, is that going to be global for Barbadense? I don't know, because we didn't take the time to pin down these homologs, because that's another entirely different path to walk down. But for Chenopodium, the paternal copy is more highly expressed. So these data can neither confirm nor refute cytonuclear accommodation in Rubisco, but it is suggesting that it is still sometimes and somewhat. So what do we make of this is the question. So given all that I've shown you, and I know I'm getting really close to time, we suggest that cytonuclear accommodation, expression accommodation, is probably subtle and or variable both among lineages and among genes. And that expression level dominance and homolog expression bias are just not general consequences, but they may be more restricted to individual cases. And that global cytonuclear accommodation just may not be necessary because in most cases, maybe the homologs are operating perfectly fine or operating equally with respect to the cytoplasm. But to truly understand what is going on in the nucleus relative to the organelles, we're going to need future work on cytonuclear accommodation that requires both additional sampling of polyploids of the different ages, of polyploids of different diploid divergences, and additional parameterizations that include things maybe like gene loss or gene conversion to kind of paint more of a 3D picture of what is going on when you take 2 divergent genomes and put them back together with a cytoplasm of only 1. So with that, I would like to thank my co-authors here and also those that provided resources in order for this work to be completed, those that provided biological resources and those providing computational resources. I'll note that this work is funded by the NSF and the USDA. And with that, I can take any questions.
All right, yeah, thank you, Corrinne. That's a wonderful talk. If there are any questions, folks can feel free to put them in the chat box or pop on and go ahead and ask either way.
Let's see, can you read that? Do I get the chat box?
You might see it. There's nothing there yet, but it usually takes about 30 seconds for folks to articulate things.
Well, I'm also, if anyone wants to ask me about AI generated art, I can do that too. This is actually, I tried putting in, I think this one here was cytonuclear, or I put like chloroplast and nucleus conflict or something like that. And it generated.
There you go. You get some H.R. Geiger, some alien stuff there. All right. Well, we have a question here from Joel Charbreau. Feel free to pop on your audio video if you want, but could there be an interaction between expression level dominance and homolog expression bias?
I mean, certainly there could be. How, I guess I did not go through and essentially see if there was any correspondence between those exhibiting homolog expression bias and those exhibiting expression level dominance. That would be actually really interesting, and we probably should have done that. Joel, who is an author, but that's something we really could look at.
Yeah, I was going to say, oh, go ahead, Joel, sorry.
We're good, sorry.
Yeah, I was going to say, I find it really interesting, and I think that you sort of touched on it, but I think it's, when I think about the sort of these kinds of analyses, we've done these and other folks too, where you've got the sort of broad classification of these categories, but it may be that the actions that we really want to see with these kinds of interactions is happening at the level of genes and gene families, and it's not. It seems that for at least some of these resources, the genomic resources still just aren't really quite there. Do you think? And as I think about the complexity of this, do you think that sort of one way to do this would be to get deep into like pangenomic data, so that you have, it's another. I know that adds another layer of complexity, but the sort of presence absence variation that I kind of wonder how much noise that adds to these kinds of analyses. And if doing this at a population or a pangenomic level might give us a way to filter out that layer of noise and get down to what might be these true signals.
Yeah, I think there's definitely something there. And, it could be too that, maybe paralogs are functionally interchangeable as well for some purposes. And, so it may not be that so a pangenome approach, looking at these in terms of gene families, that's where I really think that someone with some real good machine learning brain can really start to pull in all this information and maybe start to make a complete picture out of it. But as you say, we have to wait for a lot more resources to come online to see what trends we can find across time, across diploid divergences, across the monocot-dicot divide,
Yeah,
there's a lot.
This is very cool though. All right, well, are there any other questions for Corrinne right now? And protein complexes too. Oh yes, I see Jonathan in assembly dynamics, kinetics, and so much complexity. I keep waiting for the proteomics era. I feel like that was the heralded back in the 2000s that we'd be doing proteomics, and we are sort of, but I feel like there's a lot to digest there too.
I've got a question real quick before we switch over.
Okay.
So as I was listening to this talk, I wasn't thinking the broader angle that I think Mike and some other people were thinking. I was looking more at autopolyploids and incipient species complexes. So, there are plenty of within ploidy cytonuclear interaction systems. I'm thinking Campanula americana is one that I just know well because that's the lab I came from. But, in that system, there are lineages that have been slightly more divergent than others that are still considered the same species, but definitely have cytonuclear conflict.
Sure.
And so I'd be curious if there would be any utility to looking at some of these autopolyploid complexes rather than looking for things that are super divergent from each other.
Of course. I mean, honestly, I feel more information is better, right? Because I'm not as aware of the resources available in autopolyploids, and
that's because there's really not many. So that's challenge number one.
That was my impression.
Yeah. So, but if we have those resources, that would be great to dig into.
Right, and certainly if you have some autopolyploids where you have cytonuclear interacting genes, where maybe you can't look at it genome wide, but you can look at like a rubisco or something like that, it could add another piece to the puzzle.
Yeah, cool.
All right, well, we'll go ahead and have Corrine stop sharing so that we can get Evan's slides up there. And if there's any other quick questions in the transition. If not, we'll be able to circle back and come back to it at the end of both talks here. All right. All right, we'll go ahead and get started with the second half here of our webinar. Up next is Evan Forsythe. Evan is a research scientist in Dan Sloan's lab at Colorado State University, and he has a PhD from the University of Arizona, where he worked here with Mark Bielstein. Evan's research uses computational genomics, phylogenetics, and molecular evolution to understand hybridization, cytonuclear coevolution, and genetic interactions in plants. He'll soon be continuing this line of research as an assistant professor at Oregon State University Cascades, starting in January 2023. So congrats, Evan, on the new position. And with that, I'm going to let Evan take it away and tell us the other half of the story from the perspective of the organelle.
Thanks a lot, Mike. Yeah, it's great to see some familiar faces from my time in Arizona. And yeah, I think I'll get started here. So yeah, that I'm really lucky to have Corrinne have led off there, because I can kind of build off of what she mentioned. So the side of the story that I'm going to tell is one looking at kind of the interactions between the organelles and their interaction partners that are encoded in the nucleus. So I just want to start out. I'm going to get my laser pointer going here. Okay. I want to start out by, talking about the fact that, as we know, plant cells rely on the organelles, the plastid and the mitochondrion. And the function of these organelles relies heavily on what we call cytonuclear complexes. So by this, I mean protein, multi-subunit protein complexes, in which some of the components are encoded in the organellar genome itself, whereas other components are encoded in the nuclear genome and then imported into the organelles. So there's an example in this slide of the ribosomes, the organellar ribosomes here. And as you can see, they're composed of subunits from each genome. Now, many of these cytonuclear complexes assemble in equimolar ratios. So the stoichiometry, the ratios of these proteins is very important. And one piece of evidence in support of that fact is that, or that notion is that it comes from this study, in which they showed when you perturb this stoichiometric relationship by inhibiting protein synthesis in the plastids, and you also prevent the communication and the signaling between the plastid and the nucleus, it leads to this really severe phenotype. So it's clear that the stoichiometric balance in these cytonuclear complexes is really important for the fitness of the plant. And this is especially kind of striking, this level of communication and cooperation between the organelles and the nucleus, given that they experience very different modes of inheritance, different evolution, different mutational input and selection, and also that they rely on separate transcription and translation systems. So these are kind of create challenges for the kind of communication and cooperation between the organelles and the nucleus. Another challenge to this cooperation comes from polyploidy. In polyploidy, of course, the nuclear genome is doubled, whereas the organelles are not. So this could create challenges to that stoichiometry in those cytonuclear complexes. So some prior work has kind of dug into what happens during polyploidy. So this figure shows, this is looking at expression of nuclear genes, and they're showing the total proportion of all plastid targeted nuclear genes over all nuclear genes. And what they're showing in the plastid here is that and comparing this between diploids and polyploids. What we see in the plastid is that the ratio of the transcriptome that is kind of filled by plastid targeted genes goes down in the polyploid. Whereas when they ask the same question, but looking at mitochondrial targeted proteins, they see the opposite pattern. So there's kind of this inverse pattern or opposite pattern, depending on whether you're looking at plastid targeted or mito targeted. And another study looked just at plastid complexes. And what they're showing here is the expression for the plastid encoded RNA over the expression level for the nuclear coded RNA of its interaction partner. And they did this looking at two different plastid complexes. And what they see here is that the trends across ploidy levels are somewhat different, but also something that jumps out to me is the fact that we see levels higher than 1, suggesting higher expression of the plastid coding RNA in one complex, where the opposite is true in the other complex. So the point I want to make with these two studies is that there's no clear general patterns of cytonuclear stoichiometry in polyploids. So we set out to kind of search or ask whether there are any kind of more emergent patterns that come when looking at more widely at more cytonuclear complexes and interactions. So to do that, we'll ask kind of two major questions. So first, do cytonuclear enzyme complexes exhibit balanced gene expression? Is this can we detect evidence of this this expectation of stoichiometric balance that we expect at the protein level? And also, does whole genome duplication disrupt cytonuclear stoichiometry? So these are the two main questions that I'll ask. So I want to just kind of give a shout out to my collaborators here. I've been really lucky the last year and a half, maybe 2 years, time flies, to be part of this collaboration, working with members of the Sloan Lab, with Joel and his lab members at New Mexico Tech, and with members of the Wendell Lab. So I also want to point out that this paper that I'm describing, all the analyses, I essentially am analyzing data that were generated by the paper that Corrinne talked about today. And Corrinne is a co-first author on this paper. So I really am learning a lot through these collaborations and kind of just learning to collaborate and ask questions that I hadn't thought to ask before. So we have this system which Corrinne introduced. We've got 4 different polyploid complexes. And RNA-seq was performed to capture all the nuclear and organellar genes in leaf tissue from each of these species shown here. I want to point out, Corrinne mentioned this, but we used our ribo-depleted RNA prep as opposed to poly-A library prep. And this is really important because most organelle genes aren't polyadenylated. So any RNA-seq libraries or analyses prepared with polyadenylation will not give a clear picture of organellar expression. In fact, poly-A tails in the organellar genes are actually a signal of degradation. So this is clearly not a good unbiased view into kind of the RNA pool of the full cell, unless you use ribo-depleted library prep. So I also want to point out that we normalized our RNA-seq data in such a way that we are achieving essentially a TPM value for each gene that we're looking at. So these values are kind of normalized to the overall transcript pools. And with this data set, we are able to look at the expression of nuclear gene families. So they tend to be in what we call these quartets, in which you have, of course, the homolog from the 2 diploids. And then in the polyploid genome, you have homologs, the 2 homologs. And we can also look at the expression of organellar gene families that, again, just have 1 homolog in the polyploid because there's no genome duplication. And so I think this is a good time to make a kind of a clarification of how this study kind of looks at a different aspect than the one that Corrinne talked about. In her case, and many times she was comparing the expression between these two homologs. In my studies, we're kind of treating these as just one gene, in the sense we're wanting to know the overall level of protein or of RNA expression from these two homologs and compare that against its organellar interactors. So the first thing that we wanted to do before kind of digging too deep into polyploidy, we wanted to get a sense of what the RNA pool looks like, because this data set provides this really unique chance to look at kind of an unbiased estimate of the total cellular RNA pool in plants. And the first thing, and we performed several replicates, so that's what's shown here across 4 different species in this case. The first thing that really jumped out is that this RNA pool, the transcriptome, is massively dominated by plastid genes. That means that just the less than 80 genes in the plastid make up more than 76% of all the transcripts floating around in the cell. It's a really striking pattern. And the same is true for mitochondria, in the sense that a very small amount of genes make up a pretty sizable portion of the transcriptome, although it's not as clear when looking at this scale. And what that means is that all the rest of the genes, these nuclear genes, thousands of them, compose only 20% of the transcriptome. So this is really a really striking pattern that jumped right out at us. I had not realized that plastid transcripts were expressed at such a high level in the cell. And to kind of put this in perspective, we looked at per gene expression for these genes. So what I'm showing here is a histogram where I'm showing the expression level per gene for the plastid, mitochondrial, and nuclear genes. And this is on a log scale. And what we see is that the plastid genes and the mitochondria genes are just super highly expressed per gene. In fact, one gene in the plastid, PSBA, this is the D1 reaction sensor in photosystem 2, one gene makes up about 25% of all the transcripts in the cell. So it's really striking. So given this kind of pattern, this led us to ask, okay, what impact does this have on cytonuclear stoichiometric ratios among interacting proteins, interacting genes in the organelles? So we asked that by kind of grouping the genes into categories based on what their function is in the organelles. So we are looking at different complexes or processes that occur both in the plastid and in the mitochondrion. And what we see here is that when we look at the plastid encoded, or sorry, the mitochondrial encoded genes in the mitoribosome, for example, and compared to the expression from the nuclear encoded counterparts, we always see that the organellar encoded gene is expressed at a much, much higher level. So this when we kind of look at these data in a different way, that allows us to look at cytonuclear ratios. In this case, we are showing the expression of the organellar subunits against the expression of the nuclear subunits. What we see is, again, much higher expression of the organellar subunits. But what's also comes from looking at a figure like this is that there is this imbalance. These dots are always above the line, but there's also a really high level of coordination across complexes. So it appears that while there's massive imbalance, there's also a clear sign that this imbalanced state is tightly regulated. So it's not like it's just haywire expression of the organellar transcripts. Instead, there is some sweet spot ratio, it appears. So this is consistent with previous results. This study looks at just a few complexes in the plastid and in the mitochondrion, and looks at this pattern across several more plants. And what we see again is that when looking at a particular complex, we see that the organellar encoded subunit is expressed at a higher level. So again, this imbalance expression is consistent with prior work. And this same study actually asked this question in a different way. They looked specifically at some mitochondrial genes that have actually been transferred in some species. So it's a mitochondrial encoded gene in some plant species, but in others, it's a nuclear encoded. It's been transferred to the nucleus. And we can see from looking at this that this elevated expression in the organelles is a feature of the organelles themselves, not of the particular genes that are in the organelle, because when that same gene gets transferred to the nucleus, you see that its expression level is much lower. So the question is, are these ratios also disrupted and disrupted when you are looking at polyploids? Is there a polyploidy effect on this type of ratios? And I've got a little bit of a spoiler here myself, is that the answer is no, not really. We do not detect that. So here's looking at those same types of plots. I think I showed you just this one before, or maybe it's this one. But the point is that when we look across all 4 systems and across diploids and polyploids, we see essentially the exact same pattern. So it seems like this pattern that I described is more of a general feature of plants, and it is not at all impacted, as far as we can tell, by polyploidy. So to dig a little bit more into this, we wanted to look at the expression. We wanted to kind of look at the expression of individual genes in both the organelles and then in the nucleus. So what this plot I'm about to shows is the expression of plastid-coded and mito-encoded genes. And so what we might expect if there is some disruption by polyploidy is that we would see these organellar genes composing a smaller portion of the transcriptome in the polyploid. However, if there's no kind of sign of this disruption, we would expect these just to kind of be right down the 1-to-1 line. And that is what we see. We compare the polyploid versus the maternal diploid and versus the paternal diploid. And what we see is that we just basically the expression of plastid and mito encoded genes just does not differ between polyploid and diploid. And kind of as a bit of a control, we also compare the 2 diploids versus each other. And actually, if you are squinting and seeing any differences at all in these first 2 plots, they are less extreme than any differences you might see between the 2 diploids. So there's more of a species effect than there is a ploidy effect, it appears. So, and I just want to point out that, again, this is a pattern that we see really across all systems. So we're seeing no evidence that there's some kind of disruption of the organellar expression, total organellar expression. And so we ask the same question, but looking at nuclear genes instead. And I show them colored by whether they are targeted to the organelles or not. So I would say that if there is some sign of disruption coming from polyploidy, we would expect the polyploid to have higher, to have its nuclear genome expressing higher than the diploid, because the nuclear genome has been doubled. We might expect selection to compensate for this by selectively kind of down-regulating the nuclear genes that need to interact with those organelles, but we might expect to see no pattern at all, and that is what we see. So essentially, again, we detect no evidence that polyploidy has kind of shifted the overall expression level of nuclear genes or organellar genes. And again, this is a kind of widespread pattern across all species that we looked at. So this, so kind of how do we interpret this result, this kind of negative result when it comes to a polyploidy effect on expression? Well, I think a lot of information comes from a study generated by Matt working with Joel. So this was another product of this collaboration. And Corrinne alluded to this result as well. And what they saw when looking at the genome level, so they're not looking at RNA levels, but just genome copy levels, what they're seeing is that when you're looking at wheat polyploids, they're seeing higher levels of organelle genomes per cell in those polyploid. So it seems that when the nuclear genome is duplicated, so is the organellar genome. In other words, the organellar genome is present in more copies based on these mitochondrial markers and plastid markers. They looked at the same thing in Arabidopsis synthetic polyploids. These presumably haven't had as much time to evolve. And again, they see this same pattern. Whenever the higher the ploidy level in the nuclear genome, the higher the copy number in the organellar genome. And this is a really cool way of looking at this, is that they actually took polyploids in Arabidopsis and induced them to be haploids. And again, we see this inverse pattern in which you reduce the ploid and you reduce the genome copy number. So the results from this show that there's evidence that polyploids may fully compensate for any sort of stoichiometric or copy number differences between the nucleus and the organelle by just kind of altering the copies of the organellar genomes. And there's evidence here that this can occur on physiological timescales, so occur very, very rapidly. So if this is true, then this kind of defeats the purpose of some of the questions that I asked, because if this is the case, there's no need for an RNA level response to polyploidy, because it's already kind of been fixed at the level of the DNA. So with that, I'll kind of go over some of the kind of conclusions from my talk today. And first, we showed that RNA levels are really heavily imbalanced in favor of organellar transcripts being present at much higher levels. So, and this imbalance ratio is tightly regulated. So again, this seems to be I would say, an adaptive feature of the stoichiometric ratios at the RNA level. So this suggests that the one-to-one protein stoichiometry that we think must be achieved in the organelles must occur at the level of protein synthesis and degradation. So this kind of imbalanced ratio may have something to do with differences in the protein synthesis machinery in the organelles versus out in the cytoplasm. So then finally, does whole genome duplication disrupt cytonuclear stoichiometry? Well, if it does, this effect is rapidly mitigated by altering organelle genome copy number. So, and that is our explanation for why we do not detect any perturbation of RNA level cytonuclear ratios, because it's kind of already been fixed before we are even immediately following polyploidy, essentially. So with that, I do want to thank my collaborators and thank all of you for your attention. In addition to the folks I mentioned earlier, I also want to point to other collaborators who are co-authors on the paper that I described today. Want to thank NSF for funding, and thank you, Mike Barker, for kind of inviting me to talk today. So with that, I can take any questions. Thanks a lot.
All right, yeah, thank you, Evan. Those were really wonderful. I have some questions, but I'll hold off until give other people a chance. So if anybody has any questions, feel free to drop them in the chat or fire away.
Can I just jump in with a really quick one?
Go for it, Carl.
That chloroplast number change sounds so unexpected to me. Is that simply a cell size phenomenon, that the polyploids have bigger cells and therefore fit in more chloroplasts?
That's a really good question. I think that is one of the at least part of the cause. Yeah, maybe I know Joel is on this call and he can pipe in if he has kind of more mechanisms for how that might occur. But that is kind of my my thinking on the matter. But maybe there is more to it than that.
So now I want to say something about Carl's question, but would that mean that we would expect this to scale on sort of the cube function? And that doesn't look like what the graph showed, because if we're expecting it to expand volumetrically, but the question that I had, I think you've actually answered in the sense that we don't have an answer. So what's going on with these RNA transcripts after the fact? I mean, are they being, translated at different rates between the organelle and the nuclear genome? are we just making a whole bunch of polypeptides that get broken down for no reason, which seems, pretty evolutionarily disadvantaged, and I can't speak today, apparently disadvantageous. Do you have any thoughts on what the next step in the process is?
I think that basically what you express is basically kind of our model, is that this suggests to us, or at least we interpret this as suggesting that perhaps translation is just less efficient in the organelles. You need more RNA transcripts around in order to kind of achieve the same protein levels. So that could be part of it. It's also possible that kind of organelles do gene regulation a little bit differently. They kind of rely on protein synthesis to kind of fine tune protein levels, kind of in response to stimuli and things like that. I guess the thought that my kind of cartoonish thought there is the organelles, just kind of the way that they do it is that they keep an excess of transcripts around for everything. And then when they need a certain gene to be generated rapidly, then they kind of regulate the synthesis of proteins from those transcripts. Whereas maybe the nuclear genome kind of it relies more on transcription level regulation. So that's kind of my thoughts on how that might occur. But it is something that I will be honest, I think that following this result, this is an area that I want to read up on more and just kind of know. I can't say that I know the full scope of the literature on what is known on those types of questions, but I think it's a really interesting area to venture.
Getting into protein work, you're going to need more collaborators.
Yeah, seriously.
So very cool. Thanks
A lot.
I'll jump in here if anyone else is. I have a couple of quick questions. One, and this is really fascinating. I'm sort of curious if if this relationship has been explored outside of flowering plants. Just thinking about lineages, that how much of a constraint this could be on polyploid biology, and is this a specialty? This sort of scaling relationship between organelles and nuclear genome copies. And then the second part is, it would be really fun. Oh, sorry, good. Yeah.
Oh, yeah. So, well, I do know that this is consistent with what's been seen in mammals when you're looking at mitochondrial complexes. So there is some precedent for this, like extreme.
And I assume data for yeast as well, probably would be.
I think you're right. Yeah, I think that's mentioning mammals is not exhaustive at all, but yeah, there is some precedent, certainly. But when it comes to other plant lineages, God, I don't know off the top of my head what is known about that.
Yeah, it would be really fun. And I related that too, would just be, what happens in other lineages like the red algae or other groups with different, second with different plastids or different accessory genomes and accessory organelles, how that scales, if this is something that, there's a simple relationship with volume where things can just duplicate easily and make more copies of those organelles. Or anyway, just fascinating to think about. Lots of really cool stuff to explore.
Yeah, I agree. Yeah, I think that would be a really interesting route to go. Yeah.
Cool. Are there any other questions for Evan? All right. Are there any other questions for Corrinne? I think we had one that got answered in the chat. Well, we are over time. So we'll go ahead and wrap up. I really want to thank both Corrinne and Evan for giving us two great talks on polyploidy and nuclear, the perspective from the stoichiometry of the nuclear genome and the organellar genomes. And thank you for everyone for being here. We'll post this up later on. Before we go, I want to highlight that, as I mentioned at the very beginning, we have a special special issues of the American Journal of Botany and Apps with a call for papers that we just put out this morning on polyploid biology and methods. So feel free to check that out. It's up on the BSA website, and you can find that on Twitter now. And before we go to, just highlight our next speakers on December 7th will be the next edition here of the Polyploidy Webinar at 11 a.m. Pacific time. And we'll have Hans Becker and Brian Husband speaking to us about their work on polyploidy and a couple different dimensions there. With that, thank you everybody, and we'll see everyone in December.
Bye, everyone.
All right.